Hello, hi. Since we have completed discussion about the salient features and general account and thallus structure of Ectocarpus brown algae, we in this lecture will be seeing the asexual reproduction of Ectocarpus. In the upcoming last lecture, we will be seeing about the sexual reproduction. We have already discussed that it is a marine alga. Worldwide, more than hundreds of species are found, and in India, around 16 species are found, and that too in the western part of our country, in the western ocean part. Okay, then worldwide we have seen that predominantly it is found in the Atlantic coast. So just write a few points like this. If the question is of big marks, we discussed that it is having a erect branch, then prostrate branches. Okay, and we saw how the erect branch elongates and how the prostrate system works. We discussed that in our previous session. We have discussed about the details of the cell. It is having branch chromatophores, which is having pyrenoid bodies. That pyrenoid bodies will be storing laminarin and mannitol as the reserve food material. Each cell is uninucleate. Everything we have discussed. Now understand one more thing. It belongs to phylum Phaeophyta or brown alga, class Phaeophyceae, order Ectocarpus, family Ectocarpaceae, genus Ectocarpus. Within this genus, like more than hundreds of species are found. Now coming to the asexual reproduction details. Before that, understand a few things. Ectocarpus have two kinds of filaments. One is sporophytic filament and one is gametophytic filament. Actually, both of these filaments will be looking similar only, but it is for just for our understanding purpose, just for the illustration purpose, they have shown it like this. But in exam, we will be drawing both filaments as like this only, like multicellular branch structure, and in sporophytic filament we will show the sporangia, and in gametophytic filament we will show the gametangia. Apart from that, both of these will be looking similar, and sporophytic generation will be always diploid and gametophytic generation will be always haploid. Understood these points, right? Now, in asexual reproduction, only sporophytic generation will be taking part. Gametophytic generation, as the name indicates, it will be releasing gametes. Gametes are part of sexual reproduction, right? So, gametophytic generation or the haploid generation or the haploid, what is generation? Just filaments only. Okay, the gametophytic filaments will only be taking part in the sexual reproduction by releasing gametes. Since the sporophytic generation, as the name indicates, it is having spores, it will be taking part in the asexual reproduction. Now coming to the further details, each sporophytic generation will be having two kinds of sporangia. One is a globular shaped unilocular sporangium, then it is an elongated plurilocular sporangium. Both of these could be found either in the single branch or in either in the single thallus or in different thallus. Okay, either this could be both unilocular and plurilocular could be found in the single filament itself, or in some species you could find this in different thallus. That means in one thallus you could see unilocular, in some other thallus you could see plurilocular, whatever. This could be the type, like either it could be in the single thallus or in different thallus. Then coming to the details of this sporangia. If you are initially talking about the unilocular sporangium, the discussion should begin like this. From a branchlet, you can see this is a branch. From that, if a sub branch has to arise from here, that area, that particular sub branch will be called branchlet. So the first cell of a branchlet, the terminal or the initial cell of the branchlet from which a branch would have otherwise occurred, from the first cell, this first cell will be acting as a sporangial initial. This first cell will be transversely dividing into two. Okay, transversely dividing into. The lower cell we will call as a stalk cell. The lower cell will be calling as a stalk cell, and the upper cell will be a large cell. Okay. Now what will happen is that this particular large cell, this stalk cell will be, we are leaving it like this, it will be forming a stalk. The large cell will be doing meiotic division. This large cell, the stalk cell and the upper cell both were having a single nuclei, and since this particular sporophytic generation is diploid, both these nuclei are also diploid, right? So I am just isolating this particular cell from which this sporangia formed. Okay, I am taking it like this. So this was the cell from which this particular stalk cell got arised, and upper cell, this is the unilocular sporangium. Why it is unilocular sporangium? It is not undergoing any further division. It is having a diploid nuclei since it originated from a diploid plant. So this particular nuclei, will undergo meiotic division and will form 4 haploid nuclei initially. Okay, these 4 haploid nuclei will be undergoing multiple rounds of mitotic division, and it will be forming around 32 to 64 nuclei. And from these 32 to 64 nuclei, around each of the nuclei, a small protoplasm will be arising, and they will be converted to 32 to 64 zoospores. You can see I am taking one zoospore here. This one zoospore, you can see that it is having 2 kinds of flagella. It is biflagellated, and we can see that it is at the subapical region. It is not at the apex. Okay, it is at the subapical region. One particular flagella is elongated and it is towards the forward. It is projecting towards the front. So it is a forward oriented long tinsel flagellum, and it is pantonymatic. Pantonymatic means what? You see here, you can see hair like outgrowths in this flagella. That is why it is called pantonymatic. And the smaller flagella, which is oriented towards the bottom, this is actually whiplash flagellum, and it is acronymatic. Acronymatic means what? It is smooth. It is not having any hair like growth as in tinsel flagellum. Now coming to this particular like zoospore. This particular zoospore, since it originated through meiosis of this particular initial nuclei, because of that it is called meozoospore. This meozoospore will be swimming in water for some time, and finally it will be shedding its flagella and it will attach to a substratum. From that substratum it will be germinating and forming a germ tube like this. Before that, one thing I have to tell: once this spore release happens, these particular spores, all these spores will be covered by a mucilaginous sheath, a gelatinous or mucilaginous sheath. On contact with water, this particular gelatinous sheet will be dissolved, and then it will be maturing to this particular zoospore. That's one point you have to mention in your answer. Now, after germination, it will be releasing a germ tube, and this germ tube will be finally maturing into a haploid gametophytic filament. This is the gametophytic generation. You can see the gametangium. From this gametangium, it will be releasing gametes that we will see as a part of sexual reproduction. So this is the development and release of spores from unilocular sporangia. Now, if there is another sporangia called plurilocular sporangia in the same thallus, maybe in a different thallus, but for explanation purpose, we are showing both of these in the same thallus. In this plurilocular sporangia, similar process happens. One initial terminal cell from the branchlet area, that will be, I am taking this particular cell from the outside of the cell. Okay, at the sub-branchlet forming area, one particular cell got transversely divided into 2. One formed a small stalk cell and the other formed a large plurilocular sporangium. How this plurilocular sporangium arose? After the transverse division, one stalk cell and one larger cell. On this larger cell, there will be series of mitotic division, not meiotic division. Series of mitotic division will happen. Okay. In this series of mitotic division, it will be forming around 660 cells. Okay, this is one cell, this is two cells, this is three cells, like that. 660 cells are formed. Each cell may be called a chamber, because in each chamber, in each cell, there will be development of a zoospore. So 660 chambers or 660 cells are formed, and this will be arranged in around 20 lines. 1, 2, 3, 4, like this, 20 rows, around in 20 to 40 rows, it will be arranged. Okay. So these particular chambers or cells are created because of mitotic division and not meiotic division. So each of the cells will be having a diploid nuclei, and you can see that each of the diploid nuclei will be accumulating a small protoplasm around itself, and they will be released towards the outside. Okay, and once it is released towards the outside, you can see that it is also biflagellating nature. It is also subapically attached. The flagellum, one is tinsel shaped and other is whiplash shaped, and it will be swimming therefore around 4 to 5 hours. One point that I have missed in the previous explanation in the case of zoospores from unilocular sporangium, it was actually haploid zoospore. This actually will swim for around 30 minutes. Okay, it's a haploid zoospore. It will remain active only for 30 minutes and it will be attaching to the substratum. Then after the attachment, after around 2 to 3 hours, this germ tube will be formed and finally it will be maturing. But in the case of diploid zoospore, which is released from the plurilocular sporangium, it will be remaining actively swimming for around 4 to 5 hours. Okay, then it will be attaching to the flagellum. And you know how the attachment happens. You see this particular zoospore, both these zoospores are pyriform shaped or pear shaped. It is having a clearly differentiable anterior end. Okay, with this anterior end they will be going and hitting with the substratum and they will be attaching like that, and after attachment it will be slowly forming, it will be shedding the flagella, then it will be forming a germ tube which will be finally germinating into a gametophyte. Okay, so you can say which will be germinating into and forming a sporophyte, not gametophyte. This is the point that you have to be very vigilant about. The plurilocular sporangium releases zoospores and it will be germinating into a particular germ tube, and germ tube matures to form a sporophytic generation. Why? Because the zoospore is diploid. So diploid will be germinating into a diploid generation. What is a diploid generation? That is the sporophytic generation. Okay, this particular sporophytic generation then will be forming again unilocular and plurilocular sporangia, and again spore release will happen, then it will be attaching to a substratum and again a sporophyte will be formed. Okay, so this is the process that is happening as a part of asexual reproduction in Ectocarpus. One second. Yeah, this is the pencil drawing that I have made. I have made it more clear in paper. You can see that this is the unilocular sporangium. Okay, it burst open and zoospore got released. It shed its flagella, formed a germ tube and formed a gametophyte. This gametophyte will be further participating in the sexual reproduction, and it is not going to release any spores and all. It is going to release gamete and forming gametophytic, it is going to participate in the sexual reproduction. But from the plurilocular sporangium, you can see that this particular plurilocular sporangium got burst open and the biflagellate zoospore got released and it shed its flagella, attached to the substratum and forming the sporophytic generation, because the initial division that happened in this particular plurilocular sporangium was mitotic division, and each of the cells were diploid type, and that's why the zoospore is also diploid type, and diploid zoospore will be germinating into a diploid generation, right? That's why it is forming a sporophytic generation. Then again this cycle repeats. So this is all about the asexual reproduction in this particular Ectocarpus. This is the another kind of figure, because different textbooks and different online sources depicts it differently. You can see that it was a zygote. Okay, zygote got germinated into germ tube, finally forming this particular sporophytic generation. Okay, if it got created from after the sexual reproduction. Okay, you can see that the zygote was formed, then a sporophytic generation got formed and all. Then this sporophytic generation will be releasing spores. Then one particular spores will be forming the gametophytic filament, male gametophytic filament and female gametophytic filament. This is part of sexual reproduction that we will see later. Okay, now coming to the nodes of asexual reproduction. This slide you ignore. Okay, this is part of sexual reproduction. Just for the continuity, I have just mentioned it like this. Okay, asexual reproduction nodes is here. I have explained everything. You can see the particular unilocular sporangium releasing spores and germinating into germ tube and all. So this is for further clarity. You have to visualize this thing in mind. That's why I am attaching multiple diagrams and figures which are easily reproducible. You see that this is actually a kind of a cara-like structure, right? Having multiple branches and all. Then this is the nodes of unilocular sporangia. You can pause the video and write it. Then this is about plurilocular sporangia and its germination. So this is all about asexual reproduction. We will see sexual reproduction in the upcoming lecture. Thank you.